Hello, this is Professor Johnson, and in this video we will be looking at different signaling mechanisms. We'll start by looking at steroid signaling, then look at signaling through some enzyme-linked receptors, and finally we'll finish up with looking at signaling through G protein-coupled receptors. First of all, some things to know about steroids. So a steroid receptor is going to have this basic structure where it'll have a domain where the ligand can bind. It oftentimes will have a inhibitor that's bound here that once the ligand binds, the inhibitor can dissociate, and then this opens it up so that DNA can bind with it. The other thing about steroid receptors is they also often will have coactivators that are needed. So let's look at the signaling pathway. So the signaling pathway would begin with the ligand binding to the steroid receptor. The steroid receptor would then have this inhibitor, which is shown here in the green, dissociate. It would then would associate with the coactivator over here. The coactivator and the steroid receptor then would go into the nucleus where it can bind to the DNA. So this is a point that I want to make, that the number and the type of coactivators can vary from cell to cell. So estrogen, for example, will have different coactivators, and you may have one coactivator that's present in the uterine tissue, while another that's present in the mammary glands. And because of this, the cells will respond to estrogen in different ways. So obviously your uterus, if you have one, isn't performing the same function as your mammary glands. And this is because you have different coactivators that are altering the gene expression in the two different types. All right, that was it for steroid receptors. We're going to move on to enzymatic receptors, but there's something that I want to talk about, and that's the process of phosphorylation that we really need to look out before we get to it. So phosphorylation is the addition of a phosphate group, that's what's shown here in the yellow, to a substrate. In our case, we're going to be talking about proteins, but it can also be glucose, for example, or other carbohydrates. It's the addition of a phosphate group to it. What's special about this is that the phosphate is going to be negatively charged, so the addition of it will impart a negative charge where there is not one. For proteins, this adds a negative charge where before there was only a polar charge. It's always going to be added to a hydroxyl group. And this in part will change the structure of the protein, so it's going to alter the protein's function. So phosphorylation is key for activating and deactivating proteins, although it's not always that something that's phosphorylated will be activated or something that's dephosphorylated will be inactivated. It depends on the degree of phosphorylation, so a protein can be phosphorylated many times and where the phosphate groups are added. They can be added in different places in the protein. And all of this is going to depend. Sometimes a protein will be more active when it has a phosphate added to it. Sometimes it will be more active when it has the phosphate removed. It really depends on the protein. Now there are two types of enzymes that are going to be important for the addition of phosphates. The first is the kinases. So kinases are going to take a protein substrate and actually utilize ATP and take one of the phosphates from the ATP, so this up here is ADP, and add it to the protein. So now you have the phosphorylated protein. Remember, enzymes are catalyzing reactions, so this can actually go in the reverse reaction. So a kinase is going to be important, that it's always going to incorporate ATP into it. Phosphatases, on the other hand, don't utilize ATP. So they're going to add the or remove a phosphate or add if it's in the reverse direction, but it's going to have an inorganic phosphate that's being shuttled off in the process. So phosphatases are going to remove this. And so kinases and phosphatases are constantly working in the cell to really balance things out, and it'll depend on or it'll alter the functions of the different proteins depending on how active it is. All right, so this brings us to the enzyme-linked receptors. So remember, enzyme-linked receptors are receptors that have an enzymatic component for it. We're going to be looking at the receptor tyrosine kinases. So receptor tyrosine kinases have three domains to them. They'll have a domain that can bind to the ligand, and this is on the extracellular portion of the protein. It'll have a transmembrane domain that locks it or lodges it into the plasma membrane, and then it'll have an cytoplasmic domain that has its enzymatic activity. So we can see how this works. So the enzyme or the ligands are going to bind to the proteins, the receptors. The receptors then dimerize, so that means they come together. Once they come together, these are kinases. So they're going to use ATP and they're going to add phosphates to it. And that's what these yellow balls here will represent, are phosphate groups that are attached to it. Now, once the phosphates have been attached to it, different things can come in and start functioning. So you can have response proteins that are coming in and associated with this phosphorylated tyrosine receptors. You can have adapter proteins, and the receptor tyrosine kinases may just be sort of a bridge for this event to occur, but they can also be phosphorylating different proteins that are going to lead to a different signaling cascade. Now, one of the signaling cascades that we're going to look at are the MAP kinase cascades. So there's a number of different things that occur. So first, the ligands come in, they bind, and they'll dimerize, and we'll have the autophosphorylation, which allows an adapter protein to come in. So adapter proteins are just there. It's going to let other proteins come in and bind to it. One of these proteins is the small G protein RAS. RAS is going to get activated. We'll talk about how that happens in a second. Once it's activated, it in turn can act on MAP kinases, so it activates these. And it's going to be, there can be multiple reactions here. So remember from the previous video we talked about signaling cascades. This is the beginning of one. This RAS can activate multiple MAP kinases. The kinases in turn will activate MAP kinase. And so this MAP kinase has been phosphorylated. So notice that this MAP kinase, the kinase indicates that it's going to phosphorylate MAP kinase, which in turn is going to phosphorylate MAP kinase. And MAP kinase ultimately will go on and phosphorylate other proteins that will lead to a cellular response. So, and then each of these proteins can activate multiple enzymes or multiple substrates for them. So they can this MAP kinase can activate multiple MAP kinases. And the MAP kinase can activate MAP kinases. Can do this one, and then another one can come in. So this helps to amplify the signal within the cell. And yes, I realize that the MAP kinase is getting a bit long, but the kinases are referring to the protein that they're going to phosphorylate. So you add another kinase to the end of them to indicate that the next one is the the next kinase that's getting phosphorylated. All right, so I said we'd talk about the small G protein. So G in here stands for the GT or guanine nucleotide bonding. So over here we have ATP. That's what was shown in the earlier videos. I'm putting a gray box here. We don't need to know the overall structure. But GTP is very similar. I put green to represent guanine, so hopefully the two G's will help you keep it straight. But this has a very similar structure, but it has a different nucleotide base. So this is one of the bases in DNA and RNA, and this is a different one of them. And so you have these triphosphates that are present in the cell. GTP is going to play a distinct role from what ATP is playing. So how G proteins work, you have your G protein that has GDP, so it only has 2 phosphates on it bound to it. So this is it in its inactivating state. It gets activated by GEFs. So guanine exchange factors are what GEF stands for. These GEFs are activating the protein by exchanging the GDP with 2 of them phosphates on there for the GTP that has 3 phosphates. So you've exchanged that, and now you have your active G protein. Now this G protein can go on and activate other proteins, but also has this enzymatic activity of its own where it can hydrolyze, meaning that it's going to remove one of the phosphate groups from it. So it can hydrolyze this. When you do that, this is GDP then, because you removed a phosphate. That's what's going on here. And it returns to its inactive state, so it's no longer activating the protein. So these gaps so there are proteins known as G protein activating proteins, or abbreviated as GAPs, that can activate these proteins in the cell. And that's a nice segue into the G protein coupled receptors. So the RAS and the other G proteins that interact with the tyrosine kinases are small GTPases. The GPCRs have heterotrimeric G proteins. Hetero means different, tri means 3, and then the meric is 3 units. So it's made up of 3 different subunits. The 3 different subunits are G alpha, G beta, and G gamma. And G alpha is going to have the role that we saw earlier. So it has the same enzymatic function as before. So how G proteins work, you have the ligand that binds to the receptor. This stimulates the exchange of GTP for GTP. So the G alpha subunit and the G beta gamma subunits now dissociate from each other. G alpha goes to an effector protein. The effector protein starts spitting out the secondary messengers. It actually makes them, so it's an enzyme that catalyzes reaction. We'll look at some of these reactions that produce these second messengers that go out into the cell. Now this can all end when you have your GTPase activity here break off the phosphate from the GTP, making it GDP. So this inactivates the G alpha subunits. So the G alpha subunits reassociates with the beta and the gamma, and the beta and the gamma reassociate with the protein, where if you would have another ligand come in and bind, it would go through this procedure again. So now just looking at a few of the effector proteins, there's 2 main ones that we'll look at. The first one is adenylyl cyclase. So adenylyl cyclase, once it's activated by the G alpha subunit, is going to take an ATP. That's what this is over here. And it's actually going to remove 2 of the phosphates from it. So you have these 2 phosphates that have been removed. And it's going to make cyclic AMP. So adenosine monophosphate is what the AMP stands for, and it's cyclic because it's bound to the sugar in 2 different ways. So this is going to be a secondary messenger that can go on. It interacts with protein kinase A, and which in turn can phosphorylate different proteins and have its own role in it. So protein kinase A is going to, or cyclic AMP is going to go on to bind to protein kinase A, and then that becomes active and will produce more things in the cell. Now, phospholipase C is actually going to use a phospholipid as its substrate. So the phospholipid is going to be lodged here in the membrane. It's going to remove the phosphate head from it, leaving a diacylglycerol up associated with the membrane, while the IP3 can dissociate and move about in the cytoplasm. So the IP3 is what's made off of the polar head, and the DAG, diacylglycerol, is what remains up with the tail. And this is just a look at those structures. So over here on the left is the PIP2, which is the phospholipid that phospholipase C uses. And then this is diacylglycerol over here on the right, and on the lower right we have IP3, which is going to go on and have other actions in the cell. And that's what's going on here. So IP3 can bind to receptors on the smooth endoplasmic reticulum. This leads them to open, so these are channel-linked receptors. Once they're open, it can allow calcium to go out into the cell, and then other proteins that are sensitive to calcium are going to bind to the calcium and be activated by this, and then you'll get a cellular response from that. All right, I hope that was informative to you, and I'll see you guys in class.